This is the one-page notes of the cell cycle. And you're going to need your notes page plus 4 different color markers. And it doesn't matter what for, plus you're going to need a regular writing utensil, a regular pen or pencil. So you're going to follow along with the notes, and you can pause as needed and go back so that you are able to get all of the information. I'll be asking questions throughout, so when I do, think about the questions in your head, and that'll be a way to help you review other information as well. So we are looking at cell cycle. This is the cycle, the stages that a cell goes through to create new cells. So the cell cycle to create new cells.
An adult human contains an estimated 100 trillion cells, and yet we start life as a single cell. To grow, develop, and repair tissue damage, we rely on cell division. Every day our bodies must produce millions of skin cells to replace those lost through normal activity. Each of these cells must have a complete complement of the genetic material.
And we have 3 basic stages. There is stage 1 up here, interphase. Stage 2 has several parts to it, mitosis. And stage 3, cytokinesis. And we're going to put a little pie chart that shows the amount of time the cell spends in each stage. So let's flip it over to the back. And we're not worried about this part up here yet. We're just going to use the bottom corner. Use your regular writing utensil. And we're going to make a circle . Which is going to represent the amount of time the cell is spending in each stage here. To make my pie chart, so I'm going to label this time in cycle, and I'm going to divide it using the 2 o'clock as my reference. So it's about where the hands would be if it was 2 o'clock. So there's a big massive chunk out here. Then the remaining chunk, if I kind of visualize a little bit less than 1/4 of what's remaining, split it up like that. So it's my time and cycle. We're going to split it up. Out here, this is interphase.
And what the cell is usually doing most of its daily life, which is actually a phase called interphase, where it's growing and it's replicating its DNA and carrying out some of its cell processes. That's what it spends most of its time doing.
So we see we spend a significant time in that first one called interphase. Then this larger of the 2 remaining parts is mitosis. And the last part, the shortest, is cytokinesis. So cytokinesis, the longest name, the shortest amount of time in the cell cycle. And in cells, the interphase would be where a cell spends its life. So a cell like our muscle cell or our skin cells or most of our nerve cells, cells that are doing things and not dividing, are going to be in interphase most of their life. So if we look at our cell parts here, we have a big organelle in the middle, and then we have a cell membrane, big organelle. So what do you think that big organelle is? So if you guess the nucleus, that would be correct. So let's label that nucleus. That's just my regular writing utensil. Now pick one of your colors, doesn't matter which color, to be your hint color. So our special hint for interphase is that this is in between. So in between everything, the cell division. So in between the other two. But it turns out this is when a lot is happening. So cells will spend most of their life in that time. If they're not dividing, then three other things. I'm using my regular writing utensil. We have replicating DNA, we have growth, and other preparing to divide, which includes things like making extras of all the organelles. So we replicate the DNA first, but then all the other organelles also need to be copied so that the cell can give half to each of the new cells. Here we see a whole bunch of cells going through interphase. Alright, so let's draw some parts inside the cell. We got the nucleus, and then what's inside of that nucleus? Well, surrounding it is the nuclear membrane. There would be a nucleolus inside, and we also have chromatin. And since the nucleolus we're going to talk about more in genetics, we're going to focus on the chromatin. So I have two colors for the chromatin. And I just kind of draw them as squiggly lines. There's color 1. And there's color 2. So that is my chromatin. What is chromatin made of? Try and think on that one, and we'll come back to it. Then my last color is going to be the centriole. So if this is a centriole, that means we're talking about what kind of cell? Well, the answer is an animal cell. So the animal cell has the centriole. So these are animal cell division. Plant cells, kind of similar, but they have a cell wall to help do some of the job of the centriole. So there's our basic parts that we're concerned with for cell division. Then our next stage is mitosis. So I'm going to take my title color and right above the dotted line. So notice we have this dotted line that kind of covers all the next 4 stages. And I'm going to take this title color and kind of outline that dotted line. And I'm going to add to my little outline up here a box so I can put real big mitosis, which is division of the nucleus. So we call that nuclear division. So that nucleus, that's the control. It controls all the other organelles, tells them what to do. So without the nucleus, the cell would be in big trouble. So very important that the nucleus divides correctly so that both new cells have all the information they need to have to work. Here's a quick visual overview of mitosis in some real cells. Still using my hint color. Our hint is pairs. Is prophase pairs. So the first pairs we're going to see is that chromatin, which is made of DNA, is going to do something called condensing, which is just a fancy word to say that it's folding. And it folds up into special shapes, kind of X shapes. And this makes something we've talked about before, the chromosome.
Following DNA replication, the chromosomes condense in the nucleus of the cell. DNA condenses by wrapping around cores of histone proteins, forming nucleosomes.
In this animation, we'll see the remarkable way our DNA is tightly packed up so that 6 feet of this long molecule fits into the microscopic nucleus of every cell. The process starts when DNA is wrapped around special protein molecules called histones. The combined loop of DNA and protein is called a nucleosome. The end result is that the DNA is tightly packed into the familiar structures we can see through a microscope, chromosomes. Chromosomes are not always present. They form around the time cells divide when the 2 copies of the cell's DNA need to be separated. At other times, as we can see now after the cell has divided, our DNA is less highly organized. It is still wrapped up around the histones, but not coiled into chromosomes.
And notice those chromosomes are in pairs. So we're actually going to flip the paper over because we want to see a little bit more detail about this chromosome. So now we're on the back and we're looking at the chromosome. And the chromosome has several parts. And we start, and it doesn't matter what colors you're using for the back, each side of the chromosome, each side of the X. So this whole side here, and this whole side here, each side is an exact identical copy of DNA, because they're made of DNA. And we call each identical copy, so this is identical to that, we call each one a chromatid. So the chromatid is an identical rod of DNA, which means it's 1/2 of a chromosome. And the center part that holds the 2 chromatids together is called the centromere. So the whole chromosome is made of DNA that's been specially folded around proteins. So that's what we mean when we say it's condensed. Here's a picture of someone's chromosomes. It could be yours. So let's flip back to the front and take another look at prophase, or pairs. We notice something happening to our nucleus around the pairs of the chromosomes. The nucleus is kind of disappearing. So the nuclear membrane is disappearing. So let's label that. And the whole nucleus, I'm just using my regular writing utensil, the whole nucleus is going to basically take over the cell for this short time. And that just makes sure that the cell sorts out all this important DNA correctly. So make sure you have that copied. Nuclear membrane disappears. And we want to remember that the chromatin condensed. So chromatin condenses. To form chromosomes. Then don't forget our centrioles, or other color. The centriole, they already were in pairs, but they start to move away from each other. But they want to stay pairs. So they stay connected. These long fibers. These long thin fibers. And we call those fibers spindle fibers. Here's a picture with a few cells pointed out going through prophase at different points. So one has the nuclear membrane gone. In this picture, can you find the one going through prophase? If you guess this guy in the middle, you'd be correct. Next step of mitosis is metaphase. Prophase, metaphase. Metaphase, our hint word, so get your hint color, is middle. Looking at this picture, can you guess why? In metaphase, which is kind of the middle of the cell cycle, in the middle of mitosis, our chromosomes line up in the middle. So there's my two pairs lined up in the middle. The centrioles now are on what we call the poles, because I kind of think of it like a globe, so that would be the poles. And they are now connecting to the middle of these chromosomes. So they connect up to the centromere of the chromosomes. So, chromosomes line up. And our only other information here is about the spindle fibers. So we want to remember spindle fibers connect to centromeres. This picture is a computer rendering of chromosomes lined up in the middle, and they're so long you can see they're overlapping. That's the blue in the middle. The green is the spindle fibers. And this is just a cell under a regular light microscope, and we can see the chromosomes lined in the middle. So after the middle, anaphase. So anaphase, third step of mitosis. Anaphase, our hint is apart. So we started with pairs, but now our pairs get split apart. And it's actually the pairs of chromatids. So remember, those are identical pairs of DNA held together by the centromere. So each part of the chromosome gets split and pulled, kind of reeled in like a fishing line by are spindle fibers attached to those centrioles. And it looks pretty cool. So if you think about, you hold a streamer in the middle and you run down the hallway, and you're going to have both ends kind of trailing along behind you. That's what the chromatids look like as they're getting pulled apart from each other. So there's the chromatids getting pulled apart. And the centromeres break for that to happen. They split apart. And also notice our cell is stretching out. Chromatids get pulled to opposite ends. We call those the poles. Here is those green spindle fibers pulling the blue chromatids to opposite poles in anaphase. And once again under the regular light microscope. Last hint for mitosis is 2 nuclei. Notice something's reappeared. Our nuclear membrane comes back, but this time we're going to have 2 of them, because we're going to have 2 nuclei that both need their own membrane. So the nuclear membrane comes back, and our chromatids, they don't need to be condensed anymore, so they're starting to stretch out. Inside those new nuclei. And our centrioles are just kind of hanging out now. Their job's done for a while. They're just floating out there in the cytoplasm. So chromatids stretch out. And that brings us to our final stage of the cell cycle, and it really starts here. We can start to see it happening. Cytokinesis is cell division, so the whole cell split. So mitosis is nuclear division. Cytokinesis starts with cyto, with the C, and actually means cell. It's cell division. So our hint is cells. And at the end of this, each daughter cell is going to get half of the organelles, and they each get 1 of those nuclei that was made. And the nuclei, oops, that should be my centriole color. The nuclei are identical, so they have the exact same DNA, because remember the chromatids were each an identical copy. So inside the nucleus, the chromatin's just back in that squiggly line state, kind of relaxed out. We don't really see it. Is anything too special looking. But it does have all the information the new cells are going to need. We call those new cells daughter cells. So they get half the organelles, but all of the DNA. It's a full copy of the instructions, the DNA. The order of the steps, something that helps me remember what's going on, is I just remember that I P Mat C. So I P Mat C. And that just gives me the order of the whole thing. But I have to remember P Mat is mitosis.